And then A negative for F for A positive.
All right, go ahead and hold up your finger for the one that's B negative here. One person on your team. Do the vote. Which one would that be? B negative. Choose one person to vote there. Good. So everybody's saying C from what I can see. Okay, so these are just these are representative of red blood cells, right? And these red blood cells have the little antigen sticking off of them, right? So what do we call A? A negative, right? So the negative and the positive is 100% related to the RH factor, right? If there's no RH there, it's negative. If you do see RH, it's positive, okay? What do we call B then? O negative, right? Then we go B negative. And what's D? AB negative again, okay? What is E? AB positive, okay? F would be A positive, G would be O positive, H would be B positive. Okay, got that. Questions on that at all? Okay, so now the other thing we need to think about when we think about somebody's blood is not just the antigens on the red blood cells, but we need to think about the antibodies in their blood as well, okay? And so go ahead and talk through that. Which antibodies would each blood type have present around them? Those aren't on the red blood cell, but they're around in the plasma.
So
Yeah.
with RH,
Yeah.
because it's not technically in there until you're
So there's some kind of exposure.
so
or
there's an
something.
exposure.
Yeah,
So would you
yeah.
say that
You-
there's an antibody there?
Usually, yeah, usually it's kind of like the safe way to assume that there is an
Okay,
antibody
okay.
as far as a transfusion goes.
Okay.
I think there's a way to test, like if you only have that kind available, there's a way to test, see if that antibody exists, but yeah, normally you try to avoid that mismatch. Yeah,
Okay.
no matter what, yeah. No, you're good.
Okay, no, I was talking about B.
Okay.
Or, I mean, I was talking about B.
All right, so you're gonna be holding up the one that has no antibodies. All right, go ahead and hold that up when you're ready. Which one would have no antibodies? Okay. We got a mix of D and E. Okay, and there is a reason for that, right? The one that for sure has no antibodies would be E, right? Because if E has any of the antibodies, it's going to stick to one of those because it has all of the antigens, right? RH is unique in that we don't necessarily spontaneously form antibodies against the RH antigen. But if there was some kind of exposure to somebody else's blood that had the RH previously, then you would potentially have formed antibodies against that. And so yeah, that's why we still try to not do that mismatch of the RH factor, even though somebody without the RH factor doesn't necessarily spontaneously produce those antibodies. There's a chance that there was some exposure that we didn't know about, right? And the RH antibodies are there, right? So yeah. So E is the universal recipient, right? Because there's no antibodies there, right? We'll talk about that one here a little bit more in a sec. But questions on that so far? Okay, so then let's work through this. There's two questions here. Okay. Number one, which components of the donor's blood are actually transferred to the recipient in a standard blood transfusion? And then number two, which components of the recipient's blood do we need to be concerned with? Okay, so work through both of those questions, get your answers for them, and we'll come back together here in a sec.
So for D, what antibodies would it? then?
It could
RH.
potentially have RH, yeah. If there was some exposure to RH blood in the past.
Okay.
So say, yeah, let's say mom was RH positive, then of that person, then it would have already had that exposure. Yeah, yeah. So, yeah.
Find out.
San Diego.
All right, are you ready for the first one? Hold your hand up for the first one here. Which components are actually transferred to the recipient? Okay, let's see. Yeah. Yeah, so that, which one is 7? Is that G? Yeah, okay, most people are saying 7. That's it, right? That's it, and that's the key thing to understand, okay? Is that the red blood cells are really the only thing transferred to the person unless they're doing some different type of transfusion, like a plasma thing, but a standard transfusion is just red blood cells, okay? Because you can give saline solution to get their volume back up with the plasma, but we need red blood cells. We can't just make them synthetically and give them to somebody, right? So the red blood cells are the key component to transfer there, okay? All right, so then what about this question here, number 2? Hold your answer up for that one. What do we need to care about? What do we need to think about? Yeah, that's it, right? What we see most people saying B, and that's it. That's all we need to worry about. We don't actually care about the antigens on the red blood cells of the recipient. We need to think about them because that tells us what antibodies that person has. But the big thing is, does the recipient have antibodies that would go against the antigens in the donor's blood? That's it. That's all we're worried about. Yeah, Wesley?
Can you transfer white blood cells? That be also like they might like be programmed to attack.
Yeah, no, there is definitely some instances like that of immune disorders and things like that where there are some times where we need to have white blood cells donated, specific types in certain instances. Definitely also platelets as well. Platelet donations are somewhat normal. Plasma donations are normal for people that have blood clotting disorders because they don't have the right plasma proteins in there for the blood clotting cascade. So there's kind of all variations of different blood transfusions, but like a standard one is just the red blood cells. But yeah, yeah. Okay, so with these things in mind, yeah, you can use this. We won't do this one specifically, but you can use these questions as your guiding help for the next question, which is right here. Okay, so go ahead and work through this one with the people on your team, but use the questions on the previous slide to kind of go through step by step and figure it out. This is not something you just memorize, right? When somebody asks me, is that a safe transfusion? I have no idea. I have to work it out like this, right? Yeah, you will. Mhm.
You just hold on to your seat. I'm super excited.
That's
All right, go ahead and hold up your finger here. Is this safe or not safe? Just 1 or 2. Okay, good. Everybody's saying safe. Everybody's saying 1 here. Okay. So again, the key thing is this might be something you're thinking. I thought type A blood has anti-B antibodies. How could that be safe? yes, that's true, but the AB negative blood does not have antibodies against the only antigen that's there on the A negative blood, which is A, right? And so again, we're just working through these questions. Which antigens are there in the blood that we're giving, right? Which antibodies are present in the blood that is receiving the donated blood, right? And is there a mismatch there? That's what you need to know, right? And so you just work through that with each scenario. Again, if you just see a question like that on the test and you just try to like jump to the answer, probably not going to go well, right? Just work through those questions. It's not, I've been teaching this for 14 years now, and it's not something that I can memorize just by looking at it, right? You just have to work through the questions. So, Julia, yeah. No, no. Yeah, because that's the kind of that universal recipients. No, because they potentially have an Rh antibody. Yeah, that's the thing. So we don't know for sure, but the O negative is definitely the universal donor, right? And then AB positive is the universal recipient. Yeah, because O negative is the universal donor because there's no antigens on the blood to worry about, right? We can put that in with whatever antibodies are there. Okay. This one is safe. Yeah, for sure. Yeah. This one is safe. Yeah. So let's walk through that one more time with the AB positive blood. AB positive is the universal recipient, right? Because they can't have any antibodies there because they have all the antigens, right? So it doesn't matter what antibodies or antigens you put into their blood from the donor because there's no antibodies there to interact with it anyways, right? So the universal recipient doesn't have any antibodies. The universal donor doesn't have any antigens. That's kind of the way to think about it. Questions on that? Okay. And again, it might be something you need to work through. Go ahead, Maddie. Yeah. Yeah, I think maybe O negative, but I'm not sure. Yeah, I don't remember. Yeah. Yeah, I don't remember the exact stats there. But yeah. Other questions? You can Google it pretty quick if you want, but yeah. Okay. All right, let's do this one here. You're an EMT responding to a car accident, right? Major loss of blood. What happens? What do you do? AB negative is most common or rarest. Yeah. All right, hold up your finger when you're ready there. Okay, good. So pretty much everybody's saying C here. Isoosmotic saline solution, right? All we want to do is get their blood volume back up, right? We talked about cardiac output. We talked about the maintenance of blood pressure with the cardiovascular system. If there's not enough blood to maintain your blood pressure, you're going to die pretty quick, right? We probably on an ambulance don't have a blood supply there, right? We don't have time to figure out what kind of blood you need. So we need to keep you alive to get you to the hospital, right? So we need to get your blood pressure back up first. You can do that. You're not going to have the greatest oxygen carrying capacity of that blood, right? But it maintains your blood pressure enough to survive, right? And so that's kind of the first step. Okay. All right. So go ahead and walk through the steps here of hemostasis, starting from the top all the way through. And then just, you're going to be holding up on your fingers which number would involve TPA, tissue plasminogen activator.
That's very last. Oh my God. I think I'm never wrong. I mean, it's a compliment. I don't think that's why we were scared and you almost died.
Putting too much pressure on Alexia. She can't handle it.
Scared that she almost.
I know, I was scared too. All right, so if you're ready, if you're done filling in what these steps are, then you can hold your answer up there. Which one? There you go. Okay, I couldn't tell if you're going 4 or 5. All right, hold up your answer here if you're done, if you're ready. Which one involves TPA, tissue plasminogen activator? Okay, good. Pretty much everybody's saying 5. Okay. So step number 1 there is what we call vascular spasm, right? So we're trying to limit the amount of blood going to the place where there's a broken blood vessel, right? Makes sense. We're trying to slow down the blood flow there. Right. Excuse me. Then we get to the platelet plug next. The platelet plug, like I said in the video, is what happens after you've popped a zit and you got that clear fluid leaking out, right? How many people have had that experience? Most of us, okay. A few fortunate people have never had to pop a zit before. I don't know. I don't like you guys anymore, but I had a lot of zits when I was a teenager. But yeah, so that's it, right? It's like it's kind of leaky still. It's not quite sealed enough to stop everything, but it's sealed enough to not let red blood cells through, right? But the fluid part can leak out, right? So that's the platelet plug. Then we got phase 1, phase 2, phase 3, right? And then we got clot retraction right here. Oops. We got clot retraction, which is when we're kind of pulling that broken tissue back together, okay? And then here we've got what was that one called? Hemolysis, is that what it's called? I'm losing my terminology there, but that's when we're breaking down the clot. Is that what it is? Huh? Thrombolysis, thank you. Yeah, yeah. We're getting rid of that clot that's there. And that's where plasmin comes into play. So it's plasminogen, and then it gets turned into plasmin, and plasmin breaks up the clot, gets it out of the way. So because the tissue's healed now. Okay. All right. So that's the broad overview. Questions on any of that? Okay. All right. This one will take some time. So go ahead and walk through it. Which of the below are true? I love clinical applications in Bio 1040. So make sure you know them. Would it help if I put this slide up there? You can see that better? Yeah. Yes, it was number 5. That was the answer. Yeah, sorry.
Okay. Dear age. And then, no, that's right. That is right. And then information.
There's teams dropping like flies over here. Get on their flights. Yeah, totally. They said implant it for before class. All right, let's go ahead and walk through this. There's way too much to hold up on anything. But let's walk through it. Let me at least kind of give you the right answers. You can ask any questions if you have them. Okay, so aspirin blocks thromboxane A2. That is correct. Okay. Plavix blocks ADP from causing platelet aggregation. Okay. Next one, Plavix blocks the formation of the platelet plug, right? It does that by the mechanism previously mentioned. Okay, aspirin also blocks the platelet plug by that mechanism mentioned. Okay, and then we've got H is the next correct one. Pradaxa directly blocks thrombin. You can see that right here, right? The D there, it's directly just blocking thrombin, not allowing it to do its job, which is to turn fibrinogen into fibrin, which is that last step in the clotting cascade. Okay. All right, so that was H. Now we've got J is the next one. So Coumadin blocks the formation of thrombin. Let's look at what that means, right? So the W is where all the places it acts. So it acts in a bunch of places. So it indirectly blocks the formation of thrombin, because if we never are allowed to make prothrombin, if something above the chain of making prothrombin gets stopped, then we don't make prothrombin, and then we don't get thrombin in the end, right? So we're blocking the formation of thrombin. We're not blocking the action of thrombin like dabigatran does. Okay, what else we got? K is the next one. Blocks fibrin formation for sure. L, Pradaxa also stops fibrin formation. Heparin also stops fibrin formation. And heparin also stops clotting by helping antithrombin work more effectively. Fibrin's the clot, right? If we're going to stop, if we stop the formation of fibrin, then we stop clotting. So most of those will do that in one way or another. Yeah, Wesley?
But that was like a doesn't allow clotting and maybe I'm just wrong, but I thought that was one of the ones that like it's just through the body as you go.
Yeah, thromboxane A2 here is the only way we talked about in this class is it being a platelet activator. Right there. That's what this TXA2 is thromboxane A2. So it just stops that from binding back to other platelets and activating them. So, yeah. Okay. So just something to work through. Definitely some stuff to memorize there. Any other questions on those? Okay. Couple more here. So we'll just walk through this one together since we're running out of time a little bit here. So a clot that's stable, that's stuck there, is just what we call a thrombus. Okay, a clot that was stable and then breaks off and just starts floating down through your bloodstream. That one is called an embolus. Okay, and then finally one that was floating and then got stuck somewhere new, which is really the cause of heart attacks and strokes for the most part, right, is called an embolism. Pulmonary embolism, cardiac embolism, cerebral embolism, right? All those things are places where these clots get stuck and cause major problems for us. Okay. Go ahead and walk through this one. This is which one is not true in regards to stroke treatment? All right, go ahead and hold your answer up when you're ready. Which one is not true? Hold your answer up, please. Okay. So most people are saying B, but some are saying D. So let's talk through that. Let's talk through that. Okay, so which one is not true in regards to why it's so important to conduct a CT scan? TPA, tissue plasminogen activator, is the one that turns plasminogen into plasmin. And then what does plasmin do once it gets activated? Breaks up the clot. This is a clot busting drug. So if somebody has a stroke or a heart attack and they get to the hospital quick enough and it's discovered what it is quick enough, and they can be injected with TPA, that can save part of their heart, it can save part of their brain, right? But if the stroke they had was not a stroke from a clot, but it was a stroke from a broken blood vessel instead, you don't want to break that clot, right? That's not what you want to do, because then you're just going to cause a lot more bleeding and you could actually kill the patient, right? So you want to do that CT scan first to see if is it a clot or is it a hemorrhagic stroke. So the only one that's not true here would be B. Okay. Let me just yeah, there's 3 more slides left. We're not going to get through them all, but they will be good study tools for you. Let me just, yeah, I'll give you the answers for this one just so you can use it to study and quiz yourself. Okay, so just circle those and let me just briefly explain the last 2 slides as tools for you to study. Okay, this slide is that it just goes around the idea that all these things are proteins, right? All those clotting factors are proteins. Some of those proteins are just always in our blood floating around, and then they would be a plasma protein, okay? But then some of them are the activated version of that, so then they become an enzyme, okay? So that's what I'm wanting you to do, decide which one is which, and then where is it involved, okay? And then these ones, just making sure you understand which phase these things are found, and then what is their function. Okay. All right, guys. Have a fantastic spring break. Bye-bye. Have a good one. Yeah, see you, man. Have a good one.